phosphorylase is debranching enzyme. There's a mutase to go from G1P to G6P. And then there's G6P goes to glucose plus PI in the liver in the ER. Okay, so this is only liver, this is step 4. And then synthesis, we can say step 0 is glycogen. I don't want to call it step 0. Step 1 would be a mutase, G6P to G1P. UDP, glucose, these aren't really in order. Pyrophosphorylase, this is the enzyme that makes UDP glucose. So we kind of need this ahead of time, and we also need this ahead of time. Clearly, these three are working together. And these guys are also working together. So just so you have a sense of what the activities are. Any questions?
Does that mean that there's only a very small amount of glycogen in the form?
Yeah, you don't need a whole lot of it. Depends on how many individual glycogen particles you have, and those, you know, when you have those aggregates of glycogen, they are composed of more than just one particle. It's not just like there's one particle in the whole part of the cell that's storing the glycogen, but it's a limited number. Yeah, I don't know how many. Okay, so that's all we need to say about glycogen regulation, or glycogen pathways, in terms of how you make it and how you break it down. So we have a connection now to the rest of metabolism. Right? And we also have this specific thing about liver, where we want to send the glucose out to the bloodstream for the muscles. So regulation. So this is a cartoon of a metabolic map, simplified cartoon. All the metabolites are indicated as little dots, like here. And this is the TCA cycle. So this would be glycolysis breaking down the TCA cycle here. Amino acid metabolism connecting to the TCA cycle, and glycolysis in various ways. So you can kind of look at all these other aspects of metabolism. This is very simplified as compared to the enormous wall charts that you can purchase if you really like to spend your days, you know, looking at every possible reaction. But we can think of some general principles here, right? So biochemistry textbooks partition the material into sets of pathways. So we want to recognize straight off that inside the cell, there's no individual pathways. They're all working together at the same time. Sometimes some of them are turned on or off. Clearly, glycolysis and gluconeogenesis are not going to both be on at the same time. But what they're going on in the presence of all the other pathways, and so they're mutually influencing each other, right? So they're all linked in cells. And if we didn't have regulation, that is, if we didn't turn some of them off and turn some of them on, or at least weaken the activities of some enzymes and not others, then there wouldn't be any sort of directionality to the things, right? I mean, there are times when cells need to grow and divide. There are other times when cells may need to enter more of a quiescent state, right? There are times when cells need to differentiate. If you're starting from a stem cell and you're differentiating into different specialized cells. So, you can't drive metabolism in any particular way if you just let everything go the whole time. So clearly there has to be regulated. I mean, there's thousands of enzymes that have to be regulated, right? And the most prominent example of that would be, and the one that we spend most of our time on here, there'll be a lot of specific details so that we can see how these regulation mechanisms work in practice. But the most prominent example of, of a driving force is there's lots of energy, we're happy, we're eating a lot, we can store ourselves up, we can store a lot of extra food inside the cells, like as glycogen, for example. Or we're hungry, there's not enough ATP around, we need to do things that will conserve what scant energy we still have so that we don't die in an energy poor environment. So there's another concept besides directionality of processes of a cell. There's also this notion of homeostasis, that there are aspects of organismal function and also aspects of pathway function that require constant concentrations. Like if the glucose concentration in your blood goes much different than 5 millimolar or so, it's a problem, right? You'll have high blood sugar or low blood sugar, and there's all kinds of medical consequences for that. So that means there have to be regulatory mechanisms, not to drive things in any direction, but to keep it the same, because there's going to be external influences that would tend to impact that, and so you have to always move it back to where it needs to be. Right? If you have, in a cell, for example, if we have a pathway, say a 4-step pathway, and these are the velocities of the enzymes that catalyze these interconversions from A, B, C, D, right? If we want the concentration of B to be constant for some reason that has to do with the network of the cell at that time, then we want to keep B1 approximately equal to B2, right? So that would be an example of some kind of homeostasis on the pathway. Okay, another point is this notion of transient regulation versus permanent cellular differentiation. So most of what we're going to be talking about in here is transient, in the sense that cells are in energy rich or energy poor environments. That changes. All of a sudden we have energy, now we don't. What happens to the central pathways of metabolism that we're studying to drive them one way or another in response to this? Which enzymes are the ones that are susceptible to the metabolic cues, right? And how do they work exactly? So that's to be distinguished from something like a permanent cell differentiation, which certainly occurs probably primarily at the genetic level, chromatin structure level, which genes are turned on and which genes are not, right? How the DNA is packaged, and whether you make certain proteins or not will ultimately control whether you're going to be a blood cell or a liver cell. Okay. So these are some general ideas about regulation, why it's important. And here it's really a review. We kind of talked about some of this already. What are some of the mechanisms by which regulation can occur? And so this slide shows you 10 of them. We can rely on extracellular signals. So here we have some kind of environment. In an organism as complex as a human, of course we have regulatory signals coming from specific glands and other parts of the body that come and impact the cell, pituitary hormones, things like that. And we will see how those hormones and signaling molecules specifically interact with the cell and influence the sugar metabolism pathways. Okay. So a prominent example, for example, would be insulin, which signals particular blood sugar state. So extracellular signals could operate directly on target molecules. They could also operate, as is shown here, by getting into the nucleus in some way, either directly themselves or through some transduction signaling.